I'm Steve Darby. I'm the Associate Dean for Research in the Faculty of Environmental and Life Sciences, if you've not met me before. And it's my really great pleasure to welcome you all to what is the second of our two sets of inaugural lectures in the faculty's new professorial inaugural lecture series. So thank you all for coming to support our two speakers this afternoon. I'm not going to say too much by dint of introduction because I'd prefer for the respective heads of schools to provide their own introductions to each speaker in due course. But what I would like to do is just take a moment to explain what the motivation behind this lecture series is and was, and why it's working in this particular way. And apologies to some of the geographers in the room who might have heard this already at our first event. But there were three main reasons for wanting to set up this series of talks in this particular way. The first and absolutely the most important was just to offer an opportunity to celebrate the accomplishments of individuals who've managed to reach this illustrious position within the academic community. And I think it is really important to celebrate successes when they happen, and to give people, as speakers, the opportunity to bask in that success, and perhaps to thank some of the people that might have supported them on that journey as well. And I'm really pleased to see some friends and family members in the room this afternoon doing just that. I think it gives our speakers an opportunity to kind of share some of the things that might have helped them on their way, and perhaps that in itself can also offer others in the room some inspiration on their own career pathways as well. So that celebration and sharing aspect, I think, is a really important and valuable element of what we're trying to do here. You may have noticed, in terms of the second element of what we're trying to do with this new series, is that these lectures are explicitly designed as double headers, involving speakers explicitly from different schools within the faculty. And the idea of having two for the price of one is absolutely not some university cost-cutting measure, important though that may be, unfortunately, perhaps in the coming months ahead. But rather it's a way, perhaps a contrived way in some circumstances, but we got, I think, an interesting link between our two topics this afternoon. It's an interesting way, we hope, of bringing together people from across the community that might not normally interact with each other. So that's all of you, perhaps from the different schools and different communities within the university and more broadly. So when we get to the canapés and the reception after our speakers, I hope that there's an opportunity for ideas and thoughts to begin to engage with each other. The final element of this lecture series is that I'm standing, and I've been told to stand, and I think our speakers have been told to stand just in the box here, because all of them are being recorded. And one of the things that we wanted to do was all of these lectures are going to be placed up on the faculty's inaugural lecture series website. And over time, the aspiration is that what we'll have is a set of, no pressure, engaging, entertaining lectures delivered by a selection of our experts in our community, that as we progress through the series and build up a greater number of these talks, over time what we'll have is a fantastic resource that I hope will be accessible to anyone who cares to look into that, and can offer us an important contribution to our public engagement and outreach. and certainly looking at the fabulous lectures that we had in the first of our series, that there will be, I'm very confident, some fabulous lectures to entertain not just ourselves, but broader members of the university community and the community beyond our doors as well. So that's why we're here. Like I say, I'm going to keep this pretty short and sweet. That's really all I wanted to say, other than to express my thanks to all of you for coming along and supporting our speakers today. What I'd like to do is give the floor now to Professor Chris Horton, the Head of School of Ocean and Earth Science, who will say a few words to introduce our first speaker. Thanks, Chris.
Thanks, Steve. And to extend the welcome to you all to this afternoon's inaugural lecture here in the NOC. So as Steve says, my name's Chris Horton and I'm the Head of School for Ocean and Earth Science, and we're based down here on the waterfront campus. And in this building we have the School of Ocean and Earth Science and what is the National Oceanography Centre Southampton, and we work fairly seamlessly within the building on issues to do with marine science, climate and climate futures and the future of oceanography, as well as how humans exploit the marine environment from coastal zones right through to the deep ocean. We're a very broad school. Ocean and Earth Science gives the game away, I guess. So we have colleagues in the building who study Earth system processes, planetary science processes, geochemistry of marine environments, through to oceanography, oceanic processes, currents and distribution of water masses, through to organism biology, so the biology of the organisms in those systems. And it's a real privilege to be in a school where you have that breadth of expertise across the school. It's also challenging because we have many different conversations in the building. But that privilege is reflected today, I think, and I'm really pleased that our inaugural today, Clive Trueman, is presenting, because Clive is somebody who, for me, sits right in the centre of the school. Clive, by training, is a geologist and a geochemist, but his career is very much in marine biology. So he uses, I'm sure he's going to tell us today, geochemical signatures and isotope records to infer and understand the biology of marine organisms. And for me, that's a really good example of how our school is truly cross-disciplinary and benefits from the expertise across the school. So I'm quite sure that you're going to find Clive's talk this afternoon really fascinating, bringing together very disparate parts of marine science and earth science to understand complex systems. Just to tell you a little bit about Clive's history, Clive, from his record anyway, spent a postdoctoral research career at Smithsonian Institute, starting in about 2000. From there, he moved to Portsmouth University in 2002 to 2005 as a senior lecturer, and he came to Southampton in about 2005. So he's been with us for almost 20 years. He'll have his little 20-year badge very soon, I'm sure, from HR. So Clive, for us, is currently a professor of marine biology, and he employs stable isotope analysis and geochemistry to understand fish ecophysiology, and increasingly to understand what we call field metabolic rate, which I'm sure is what he's going to talk to us about today. So without any further ado, I'll hand you over to Clive to hear what he has for us this afternoon. Clive.
Fantastic. Thanks very much. Thanks very much, Chris. Thanks, and also thanks, Steve, and to the faculty team for organising these, setting these lectures up again, and organising and facilitating our chance to present the work that we're interested in, and to thank people that have helped us along the way. So in that realm, I'm going to go straight into saying thanks. If this works. Does this work? Yes. So you obviously have to thank, before anything else, have to thank my parents. My sister and I were first generation university students, or people going to university. I've been told to keep still. And my parents were really supportive all the way through of having an academic interest irrespective of the challenging career prospects that might hold. And so they're always encouraging strange interests and not judging, at least not vocally. So thank you so much to them. And then, of course, thank you to my family, Sally and Calvin and Meredith, who have to live with the difficulty of living with an academic and picking up dead fish from family beach visits and all those kinds of things. So thank you very much for supporting me through this. Okay. There's always a huge number of people who you rely on that have shaped what you do and allow you to do what you do. So, I don't know the latest point. So, people who are really instrumental in showing me how to be a scientist as my principal mentors as I was learning to be a scientist. These people are fantastic, and Martin's here. I'm not sure if he's here in the room, but here as well. Followed him around a little bit. Thank you to all of those amazing people. Colleagues who I've worked with repeatedly over the years, and shared ideas and have shaped the kinds of ideas that I've developed and we've developed together. Amazing people, all of them. Are fantastic analysts, analytical specialists and technicians, without whom the kinds of work that I do and lots of people in this building do would be completely impossible. These people are astounding, amazing people. Thank you to all of those, and a particular shout out to Tina there. And then PhD students. So I've been enormously lucky to have worked with, supervised quite a large number of PhD students so far. And honestly, I think this is, for me, probably the most enjoyable part of the whole job, is working with PhD students, seeing them learn how to become scientists, and then watching them flourish and progress subsequently. So I am going to present quite a lot of ideas that have come through these PhD students' work, and I have to apologize for them for misrepresenting anything, but that's the most fun that we've had, is working together on their projects. So, okay. So, my dad came from the Isle of Wight, and we would often go to the Isle of Wight on family holidays. And I think when I was about 6, wrong way round. When I was about 6, we stumbled across this relatively unprepossessing, but to a 6-year-old boy, fantastically, satisfyingly big ammonite fossil. And that effectively sparked my interests and kind of shaped the rest of my life, really, getting me excited and interested in natural sciences, geology, and also marine biology. And I spent quite a lot of my childhood torn between, did I want to do geology, did I want to do marine biology, and as Chris alluded to, kind of found a way of doing both, and perhaps exploring that those divisions don't really exist. Subject divisions don't have to be divisions, and that's kind of a theme to what I've done throughout my career. So I went to Bristol to study geology, and I did geology undergrad and PhD, and for my PhD I was studying dinosaurs, particularly dinosaur bone chemistry. And then I moved to the Smithsonian in DC, and my study area moved a little bit more recent and started looking at hominid evolution sites in Kenya, and also bones in Kenyan game national parks. I then moved to the Weizmann Institute of Science in Israel, and my science moved younger again, and we started looking at scientific archaeology and taking instruments out into archaeological digs. I think that's me, just there. And archaeological digs, looking at site formation and destruction processes. And then I moved back to Britain and moved to Portsmouth, where I had a research aquarium. And that started my interest in modern fishes and modern animals. And that's kind of where I've been ever since, is looking at marine animals. Now, this seems like a really disparate and disjointed set of research subjects. But there is something that links all of those together. Because in every area, we've been trying to ask the question of how do you study a process that you can't see? And you might not be able to see it because it happened in the past, or you might not be able to see it because it's on an elusive animal. But geologists and your geological training is all about trying to measure things, trying to study things that you can't observe directly. And so geologists and having a geological training really sets you up to think about alternative ways, proxies, for studying things that you can't see directly. And that training in finding proxies, finding indirect measurements of processes that you're interested in, that training really sets me up for the kind of work that I do in looking at obscure behaviours or behaviours that are hard to observe because they're under there. And I think just as exciting as trying to understand what happened in the past, trying to understand what's happening there in that perplexing blue, that's really what makes me excited and interested. So what I'm going to talk to you is the work that I've been doing with all of my colleagues, trying to find proxies, geochemical tools to understand animal behaviour in the sea. Okay, so we need to meet some of the principal characters now that we've established the backstory. And the main thing that we're going to talk about are stable isotopes. So we're going to use isotopes as our tracer, our signal for studying things that we can't observe. All we need to know about, for the purposes of this talk, is that stable isotopes are atoms that have the same number of protons, that defines what the element is, but a different number of neutrons. And what that means is that these atoms do the same reactions, form the same compounds, but they have a slightly different weight. And that different weight means that those reactions happen at slightly different speeds for the two isotopes, produce bonds of slightly different strengths. The upshot of that is that every reaction creates a slight difference in the ratio, the relative abundance of these two isotopes. Every single reaction involving, in this case, carbon, has a chance of producing a signal, a change in the isotope ratios. If that change is predictable and we understand it, then we can use the expression in isotope ratios to tell us something about the reaction that's happened. And really what we do as isotope geochemists or isotope scientists is try to understand how to read the story that's written in the isotopes. Okay. Lots of elements have stable isotopes that we can work with, and we're really lucky that all of the main building blocks of life have isotopes that we can easily measure and use to infer aspects of biology. So carbon, oxygen, hydrogen, nitrogen, sulfur, all of these provide traces, provide a kind of rich palette to paint a picture of how these animals live and work. And because carbon, nitrogen, sulfur are present in just about every tissue that just about every organism produces, maybe not, well, carbon is at least, we can use almost any tissue from almost any organism to study with our isotope tracers. So that's the world we live in, the world I occupy. So I'm going to ask, I forgot to say, I'm going to take you through two main questions that we use isotopes to explore today. And those questions are, where do you come from? A little bit of what did you eat? And how much do you eat? So those are the questions that I'm going to try and explore using isotope tracers. So here's my first question. Where are you from? Or where have you been? If you send me this postcard, I've got a pretty good idea that you've probably been to Paris. The reason I know that, or the reason I think that's likely to be true, is that I've got a mental map of the probability of you picking up a postcard of the Eiffel Tower in anywhere on Earth. And my mental map says it's much more likely that you're going to pick up that postcard in Paris than anywhere else. I might not be making that mental leap in my mind, but that's kind of what I'm doing. I mean, you could be picking up an Eiffel Tower postcard in Calais. There's a possibility you could be doing that, but it's much more likely that you'll get one in Paris, unless you're really obtuse and being mean. But we can use that same logic, that same idea, to try and understand where wild animals have been foraging. If we can map the distribution of natural tracers that get incorporated into their tissues. So, in the ocean, phytoplankton, plants in the ocean, fix carbon, just like plants on land do. And when they fix carbon from the atmosphere, those carbon isotopes get fractionated. They change in their relative abundance. The amount of fractionation depends on things like temperature and the growth rate of the plankton, the species of plankton. What that means is that if we understand that fractionation process, then we can build maps or models of how the stable isotope composition of plankton will vary across the oceans. So we spent quite a lot of time doing exactly that, mapping the spatial distribution of isotopes, and these are all carbon isotope maps, at different resolutions and in different parts of the ocean. So these maps then give us the signal that we can relate where an animal has been feeding to its isotopic composition. So I'm going to give you a couple of examples of how we use that to study animal movements. So, I mean, you might as well start with sharks, you know, cool marine animals. So one of my PhD students, Chris Bird, spent a lot of time thinking about shark movements and how different kinds of sharks use the global ocean. So he looked at shark isotope data from two main groups of sharks, from coastal sharks, actually you probably count this one as a coastal shark as well. So coastal sharks, sharks that inhabit our shelf waters, and oceanic sharks. And he wanted to look at how does the isotopic composition of these sharks relate to the place where they were caught. The question is, do these sharks feed, and remember they're getting their isotopes from their food, so do these sharks feed in the same place as where they were caught? So this is what he found. So to look at these plots, we've got distance from the equator on the x-axis here. So the equator is 0, it's getting further away from the equator as we go in this direction. We've got the isotope, carbon isotope values on this axis. Black dots here are the plankton isotopes in the places where the sharks were caught. And here you've got the shelf sharks, and their isotope compositions are in grey. So what you can see is that the trend of carbon isotopes that we find in shelf sharks really mirrors the trend in our carbon isotopes that we get from plankton in the places where they were caught. So that's telling us that these shelf sharks forage and feed in the same places where they're caught. For the oceanic sharks, we see something quite different, where the isotope compositions of their tissues really reflect a very limited latitudinal range between about 5 and 20 degrees north and south of the equator. Now this really matters because this helps us design ways of protecting sharks. If we want to conserve sharks, which are globally threatened in lots of different places, if we're trying to protect shelf-dwelling sharks, then creating marine reserves in the local area is likely to conserve those sharks because they are feeding and spending all their time in that local area. On the other hand, creating a regional marine protected area at, say, 35 degrees latitude might protect sharks when they come in to breed, but it's not going to protect the areas where they actually feed. And so this helps you design conservation strategies for different types of organisms. And these are organisms that are really quite expensive to track otherwise. Okay. So another example, and one a little bit closer to home. So UK, we have some of the most important seabird colonies globally. And these seabird colonies face a huge amount of threats. We have threats from avian flu at the moment. There's also threats from climate change and from competition for their food from industrial fisheries. So to conserve these globally important seabird colonies, we really need to know quite a lot about how they use their local environment. Now, just off of sort of east of Edinburgh, there's a place called the Isle of May, and this is a really important breeding colony for seabirds like puffin, razorbill, and guillemot. They winter on this island, and some years are very bad for the overwintering survival of these birds. Other years are quite good. But the same years might be good for one species and bad for another. And understanding why birds do well or badly in particular years is quite challenging, it's quite confusing. So one of my PhD students, Katie St. John Glue, spent some time initially building an isotope model of UK shelf seas so that we could start to infer something about movement and feeding from birds. And then spent some time working with people on the Isle of May to explore the isotope composition of puffins and razorbills. So I'm going to really rush through what she found. But essentially, these blobs represent the areas that the birds were feeding in as inferred from the isotope composition of their feathers as they returned back to the breeding colony. In years where there was quite good survival, puffins and razorbills essentially feed in the local area, or local to the breeding colony. And they both fed on quite high quality food. We can also infer the kinds of food that they ate from their feather isotopes. But in bad years, in years when survival was fairly poor, the puffins were foraging in the same area, but had much lower quality food. Whereas the razorbills foraged far more distantly and managed to retain the quality of their food, although presumably burnt a lot more energy getting to those distant grounds. So essentially what we're able to do then is identify different strategies that these two different birds use in years where feeding resources are limited. So what essentially happened in the bad years is that one of the main sources of food, kind of snake pipefish, was missing from the local environment of the breeding colony. The puffins were not able to move further away and so had to eat worse food. The razorbills were able to move to find their best food. So this kind of data allows us to really unpick the strategies that different species use to cope with environmental challenges. Right, that was a single postcard. What if you send me this list of postcards? Obviously, I know that you're keeping in touch really well. But if I get this list of postcards, I've now got a pretty good idea that you're having a nice holiday, but also I've got an idea of where you've been, and if I've got these in order, I can have a pretty good snapshot of the route that you've taken. So, if I can take, instead of just postcards, serial samples of a tissue that grows through time, then I can use this kind of logic to start to reconstruct movement pathways and migration routes. My prop. So here we have, this way up, here we have whale hair, or whale baleen. Whale baleen grows, it's hair, it grows just, not like my hair, but it grows from this side to this side, grows relatively slowly. This piece of hair will have grown over about 4 years. It's fin whale hair. But we can, you probably can't see from here, but we can take little samples across this baleen hair plate and build a list of postcards of the carbon isotopes that the whale was feeding on as it grew that hair. So if you've been to the Natural History Museum, you've seen this whale skeleton in the foyer, Hope, the blue whale, who died in 1891, stranded in Ireland. So we were very fortunate to work on baleen plates from Hope. And what we did to try and understand the most likely movement pathways that she used in the years before her death, we took our isotope model, carbon isotopes model, which has time and space contained in it. So this tells us what the carbon isotopes are likely to be at any time in any place in the global ocean. We then built a movement model. We basically put some simulated whales. We made some sim whale models. And we instructed a simulation to make possible whale migration routes. So what you're looking at here is one simulated whale track that's producing a possible migration pathway. And we can overlay that on this model, and that will produce a likely baleen isotope trace that would be produced by this particular set of movement pathways. We could do this 10,000 times and produce 10,000 possible migrations and the isotope records that would be produced by those different migrations. And this is what you get. Here's the best fitting 10% of those simulated migration paths. The red is what we actually measured on the baleen track. And then these blue dots represent where Hope was most likely to have been in any month in the 5 or 6 years before she died. Firstly, I think it's quite amazing that we can do this. It seems relatively trivial to try and do this for a single whale that died 100 years ago. But one of the reasons for doing this is as a scientific communication tool. Millions of people see this skeleton every year. And what we try to do is find a way of linking the bones, the skeleton, to an actual living animal and helping people to recognize that what you see as a bone was a moving, interacting animal exploring its environment. We are also using the same approaches to look at whale behaviour while there were still quite a lot of large whales in the Atlantic Ocean before they were hunted out. Now, whales are slowly beginning to recover, and we really need to know how whales used to use the Atlantic Ocean so that we can help to manage likely interactions with things like shipping and fisheries as their populations begin to recover. Okay, so that was movement. The last bit I want to talk about is food and feeding, and how much do you eat? So energy and metabolism, this is the fuel of life. What you need to live is to eat, to produce energy. And we can summarize metabolism in this kind of GCSE, maybe even before that now, GCSE equation, where basically food is burnt by reacting with oxygen to produce energy and carbon dioxide. This is how we respire. But hopefully, now you're all tuned in to say, well, these are all elements that we have stable isotopes for. And so we should be able to explore all aspects, or lots of aspects, about metabolism and respiration using stable isotopes. And for the last 5 or 6 years, this is really what I've been exploring, is how can we use our stable isotope tracers to tell us something about energy use in marine animals. So I can introduce you to another amazing thing. This is an otolith, an ear stone. So this is a small stone, calcium carbonate structure in a fish's ear. Now you need to know lots about fish ears. They're amazing things. But all we really need to know is this is like the Babel fish, Hitchhiker's Guide to the Galaxy, the thing that's just too amazing to be real. These stones grow throughout the fish's life, and so you can use them to count how old an individual fish is. This mackerel, I'm guessing, was about 6 years old. But they're made of calcium carbonate. And again, we've got carbon and we've got oxygen that we can use as some tracer for metabolism. So the oxygen in an otolith. The oxygen in the otolith comes exclusively, pretty much, from the surrounding water. So the isotopes of oxygen are fractionated when you go from the water into the mineral. Lucky thing here is that the extent of that fractionation is dependent on temperature. That's fabulous. That means that if we know the isotopic composition of the water and the otolith, then our oxygen functions as a thermometer. And the oxygen isotope composition of an otolith is telling us the temperature the fish lived in. As fishes are ectotherms, cold-blooded, the temperature of the water is the same as the body temperature of the fish. The carbon in the otolith, and this is really what we've been doing in my group for the last little while, the carbon in the otolith is a mixture of carbon from the seawater, and carbon that's released when the fish burns its food. The relative proportion of these two sources, it turns out, is directly proportional to the rate at which this food is being burnt. And the rate at which the food is being burnt, that's metabolic rate. So our carbon isotope composition of the otolith, if we know how to read it properly, is giving us a natural Fitbit. It's telling us the metabolic rate that the fish was running at while it was growing its otolith. So, all we have to do then is stick some fish ears onto a slide, drill off the powder, place it into an exciting machine upstairs, and we have a record of temperature the fish was living in, the metabolic rate it expressed at that temperature. And this is something that we've never been able to do before for wild fishes. So knowing the energetic cost of living in the sea, it's a really area that we've really wanted to have information on, haven't had before. So we've been building a database to understand how the energetics of wild fish work and vary. So we've measured a lot of animals now. I'm going to gloss over this one slightly, but we've looked at quite a large portion of the fish phylogenetic tree of life to explore how metabolism varies across these different groups of fishes. And if we know how the metabolic rate varies, from that we can also work out feeding rates. So I'm going to kind of show how this relates to other kinds of animals. For air-breathing animals, we can use a different technique to understand the energetic cost of living, field metabolic rate. And compilations of the energetic cost of living for birds in red, mammals in orange, and reptiles in green. This is pretty much the sum total of field metabolic rate data that we have for most terrestrial vertebrates other than humans. It's really expensive to do this kind of measurement, but it doesn't work for aquatic animals. But you can see here how it's relatively more efficient to be an ectotherm than an endotherm. You use less energy for a given body mass being a reptile than you do being a mammal or a bird. So now we can put fish onto that plot. So this is now all of our new data, the first view, really, of the energetic cost of being a fish. And we can see that fishes have almost the same energetic costs of living as terrestrial reptiles. But one thing we'll find is the growth rates of fishes are significantly higher than lots of these equivalent reptiles. So this is really exciting for thinking about how evolution shapes metabolism. But try and put it into context of us. So we're zooming in on the big end of fishes now. And if you're a kind of average-sized adult human, a sort of me-sized adult human, your metabolic rate is almost the same as that of a bluefin tuna of the same size. So here we are. My metabolic rate is pretty much the same. My energetic costs, the amount I need to consume in a day, is about the same as a bluefin tuna, the same size and weight as me. That's the energetic cost of me sitting at a desk, compared to the energetic cost of an animal swimming at 50 miles an hour, max top speed in water, and growing to my size in 5 years. So tuna are much more energetically efficient than humans. But of course, that's the energetic cost just of my food. I'm actually burning a lot more energy than that in terms of all of the electricity and everything else that I'm using. So the average energy use of a human, domestic energy use of a human in Britain is all the way up here, taking the heating and the light. It's not including holidays. There's one other thing that I had. How many people here use either ChatGPT or Copilot or anything like that? Okay, right. The energy cost, I had to do this several times to check I was right. The energy cost of a single query, a single ChatGPT query, not training the model, but just running the query. This is the energy cost of a single ChatGPT query. It's equivalent to the energy used by 50 grams of fish for a whole day, just for one query. It's an amazingly energy intensive thing to do. Anyway, I had to look at that. I had to do that lots of times to be sure. It really is. So you could keep a fish about that size going for a day on one query. Okay. This is basically the variation in energy costs of fishes for their daily energy needs. And the last plot really kind of compressed everything in. This slide is there to show that there's an enormous range. I don't really have time to talk about how exciting it is to have this view of how fishes work. But really we have kind of a continuum that on this side, you've got fishes that are evolved to cope to maximize all the resources that are available. So they've got really high energetic costs that allow them to make maximum use of energy available and oxygen. On this side, so this is kind of like your Ferraris of the sea. On this side, we've got fishes that are adapted to make use of low resource availability. So if there isn't much oxygen or isn't much food, then you evolve this kind of strategy. It's kind of the Hyundai i10 strategy. So we can start to understand how fishes have evolved and how their metabolisms have evolved in concert with the environment and how the environments have changed over time. I'm really excited to explore what we can learn about the co-evolution of life and planet from these kinds of data. All right, coming to the last thing that I'm going to talk about. Remember I said we can get temperature from otoliths. We can get the temperature of the fish we're living in. So if we measure a whole bunch of fish, we can build a distribution of temperatures. So we can fit a curve to that, and we get a sort of peak in that curve, and that tells us the temperature that those fish, those individuals, prefer in the wild. From the same measurements, from the carbon isotopes, we get the metabolic rate of those individuals. So we can get a measure of the metabolic rate as a function of the temperature. And we can now see that there are some temperatures where metabolic rate gets increased, so this is beneficial, but there'll be a peak temperature. And if you get beyond that peak temperature, then the fish is no longer able to cope with the extra energy demands associated with that temperature rise. So that's a limiting temperature. And with this kind of information, we can really start thinking about how fishes are likely to cope with climate change and warming. So my last example is, again, back to bluefin tuna. So here we have bluefin tuna, Atlantic bluefin tuna, and these are spawned either in the Mediterranean Sea or the Gulf of Mexico. Exactly. So over the last 30 to 40 years, we've seen a really big decline in the proportion of adult bluefin tuna in the Atlantic that had a Western or Gulf of Mexico origin. So these are fish from a Gulf of Mexico origin, the proportion of fish from a Gulf of Mexico origin over time. You can see that this is going down and going down and going down. So something is happening in the Gulf of Mexico that is not good for bluefin tuna. So what might that be? Well, we had, working with tuna scientists from the Mediterranean, we managed to assemble a very large data set of tuna otolith isotopes. And here we've got temperature from the otolith on this axis and metabolic rate from the otolith on that axis. And you can see this kind of curve that I was showing you before, where metabolic rate increases until a certain threshold temperature, at which point additional warming produces a decline in metabolic rate. Should have said the red ones are Mediterranean origin, blue is Atlantic origin. So from this we can infer that the limiting temperature for bluefin tuna juveniles is about 28 degrees. We can then go to climate models and say, when, under different emission scenarios, when will summer temperatures get warmer than that limiting threshold 28 degrees? It's already warmer than 28 degrees in the Gulf of Mexico in summer. At the moment in the Mediterranean, only really in the far eastern Mediterranean do we pass the 28-degree threshold regularly over the summer months. But under different emission scenarios, you can quickly see that we start crossing that threshold quite dramatically. And this is kind of the colour represents what year we start really crossing that threshold. So under pessimistic emission scenarios, pretty much the whole of the Mediterranean basin crosses this threshold within 50 to 70 years. What that means for bluefin tuna is that they'll effectively be pushed out of the Mediterranean, and spawning and juvenile growth will start moving north into places like the Bay of Biscay. At the moment, Bay of Biscay has really big pelagic fisheries for things like anchovies and sardines. Bluefin tuna juveniles will get caught in those fisheries. So if we want to have thriving bluefin tuna populations in the future, then we're going to need to start thinking about how do we monitor and regulate the pelagic fisheries in the areas that tuna are likely to be pushed into. So it's a way of having some foresight to develop mitigation scenarios before we need them. Okay, so where do we go from here? Well, the next little bit of research, we want to carry on understanding how metabolism moves across or is distributed across different fish populations, how fish adapt to climate, and what that means for the coevolution of fishes and the Earth. At the moment, my data set is really dominated by cold water, temperate fishes. So I think we're going to have to go to the tropics. Is that okay? We really want to have more use of the kinds of proxy data that we're generating used in management and policy. So we're going to spend quite a lot of time talking with fisheries science agencies and explaining what potential these kinds of techniques can deliver for management, and also how some of these techniques are actually quite cost effective compared to doing long survey campaigns on boats. And finally, so I'm going to start going back into the past and thinking again about evolution and what is the response of life and physiology of life to big changes in climate and climate stability. So these are areas that we want to explore. So I just finish on a kind of little note that I think it's amazing to me. It's still amazing that we can take something as mundane as an earstone from a fish and extract the temperature it was living in, where it was living, what it was eating, and how much, all from just making some measurements on this small bit of tissue. And I think that's kind of an expression of the human ingenuity and scientific creativity of hundreds of people that allow us to be able to find ways of doing these kinds of measurements. And that's really the thing that keeps you going, the thing that makes science so much fun. And it's an enormous privilege to be able to have chased those ideas for as long as I've had to do it. And I know how all of this stuff works, more or less, but I still think it's a little bit magic. And I hope I always do. Thanks very much.
Okay, thanks, Clive, for a really fascinating tour de force of the work that you've been doing. We've got time for a few questions, if there's any questions from the audience. Stony silence. Justin.
I think Clive at the beginning
I can hear you.
Okay. I think at the beginning you said something about you were going to talk about some of the obscure things that you found. Was that right?
Oh, no, I think, sorry, I might have, I was really thinking about fish behaviour in general as being a little obscure.
Oh, okay, so the topic's obscure. Well, let me just follow on and say, is there anything that you found in your research that was really surprising that just took you...
All right. Yeah. I mean, almost every analysis gives you something that's interestingly surprising. So one of the things we've been working on is, at the moment, are deep water fishes. So the most abundant vertebrates on the planet are little tiny fishes that live typically around about 300 to 500 meters below the surface. And what we've been interested in what the role they play in moving carbon from the surface to the deep. There's lots of different kinds of these fishes, but one of the species that we've been looking at, which is actually the most abundant vertebrate on the planet, as far as we know, more abundant than humans, more abundant than any other vertebrates, we discovered they have almost no metabolic rate at all. They've got incredibly low metabolism. And to the extent that we thought, our methods must be wrong, this can't be right, they can't have effectively unmeasurably low metabolic rates. So then we started looking at their gills, and their gills have almost no gill filaments, a thing that you use to get oxygen out of the water, and their muscles are almost completely water. These animals are incredibly efficient, and they've adapted effectively to have, to be able to survive on tiny, meager particles of food that sink down to that depth, and also to live in oxygen minimum zones. And it's incredible to see just how adapted, how flexible the body plan of fishes can be to make use of those kinds of environments.
Any other questions for Clive? Lisa?
Thanks, Clive. Lovely talk. Is that on? Yes.
Yeah.
Going back to your beginning studies where you were looking at the carbon isotopes with your whale and your bird feeding ground example, I mean, looking at the global maps and those Northwest Europe maps of the isotope values, they're clearly non-unique. So do you use other data to narrow down the scenarios?
Yep.
I mean, you showed that a bit with the whale, as in you generate multiple scenarios as to where it would be. But I was thinking particularly with the birds, do you have sort of tracking data that enables you to home in on where they've gone?
So yes, absolutely. I only showed one isotope map to make it clearer, but we use 3 different isotope maps to have 3 different kind of palettes. So we have carbon, nitrogen, and sulfur for the birds. But we also do have some light-based geolocators on those birds as well. So in that study, we can use the light-based geolocators that have quite a coarse resolution, and we can use those as priors in a Bayesian model to identify to narrow down the likely feeding areas. Yeah.
Any final question for Clive? If not, I'd like to thank Clive for what has been a fascinating talk this afternoon. Hopefully he's given you a flavour of just some of the types of work that we do here in Southampton, and that we're really privileged to have people that think broadly about science questions and bringing together different techniques to apply to these different kind of really difficult systems. And I think techniques that, as time goes on and we're looking for ways in which to be more efficient with the data that we collect, are going to become more and more important as we try and understand how organisms survive on planet Earth, ocean Earth, but also how things are going to change in the future and how humans might impact those things in the future. So if we can show our appreciation for Clive one more time.